The symptoms caused by the bacterial infection can be necrosis, hypoplasmia, hypertrophy, wilts, dumping of. All viruses are obligate intracellular parasites that do not have the molecular machinery to the replicate without a host. Viruses are not active outside of their host. Viruses cause the many important plant diseases and are the responsible for the losses in crop yield and quality in the all parts of the world. Chlorosis, yellows, necrosis, ring spotting, destruction, stunting, breaking, and the greening of the blooms are few important symptoms produced by the virus. Some of the bacterial diseases, necrosis and death or killing of the host tissues induced by the attack of the pathogen is also called as necrosis. It may be caused by the rots, blights, wilts, and dieback, and cankers, and may be general, local, gradually, or rapid. When the necrosis generally is also called as the decay or rooting. The causal organism may be bacteria or fungus rooting in invertebrate plants. Roots are the many kinds. The common ones are the soft roots, dry roots, wet roots, hard roots, brown roots, and the black roots. These roots cause the general necrosis. Local lesions or spots, mainly almost all the bacterial leaf spots and the blights are caused by the genera Pseudomonas and Xanthomonas. Mainly spots and these are the caused by the Pseudomonas species, some of the Xanthomonas species. Blight, in many cases, the leaves, the stems are fixed in response to the attack of the pathogen undergo rapid discoloration followed by the death. The dead parts of the become dark or brown in color. This condition of the affected organ is also called as the blight. Soft roots. Soft roots attack a larger number of hosts and they are the best known of the causing disease in fleshy plant structure, both above and the below ground. Organism responsible for the soft roots reduce the plant tissues of a soft, very moist pulpy mass of the bacteria produce the enzyme that break down the middle lamella and disintegrate of the cells. Rooting tissue becomes the watery and the soft of the bacteria will form slimy pole smelling liquid that the ojos out in the infected tissue. Bacterial ojo is the diagnostics of the soft root diseases. The bacteria responsible for causing the soft roots living plant tissue include the Erwinia species, the Pseudomonas species, Bacillus species, Clostridium species. These are the four species are the caused by the soft roots, mm, especially rots diseases. Next one is the canker. Canker is a dead lesion. Cankers are the crocky outgrowth of the form on the trunks, stems, twigs, and the branches, and all other the above ground plant parts. Cankers can be slightly sunken and dark brown and much longer than the broad. The cortical tissue of the canker can be orange brown to dark brown. Gum is produced in most cankers and some branches and the twigs. Cankers that do not produce the gum may have the sour odor and the be soft, sunken and moist. Cankers with the ridley trunks and the branches can result the leaf stress and even to the portion behind the canker dies. Mainly the cankers also it belongs to the Pseudomonas and Xanthomonas cause the canker diseases. Xanthomonas citri, Pseudomonas, tumors and galls. Some bacteria produce the galls and tumors in plants. The abnormal and the disorganized the growths are the produced by the action in organism and the meristematic tissue of the plants. It results in the destruction, swelling, leaf galls and galls. The excessive growth may be due to the two processes, hyperplasmia and hypertrophy. The former results due to the rapid cell division and thus increase the number of cells of the plant tissue. Hypertrophy leads the abnormal increase of the size of the cells holding. In many bacterial infections, rapid and continuous cell division, hyperplasmia and hypertrophy of the plant tissue takes place. As a result, galls or tumors develop on the affected organs. Galls have been referred to as the crown gall, crown knot, root knot, and root gall. Galls first appear in a small, whitish, soft, round growth of the typically on the plant's crown. The main root. Bacterial galls can be produced in the genus Agrobacterium tumefaciensis in the Agrobacterium ruby, Agrobacter rhizosinensis, certain species of the Arthrobacter, Pseudomonas, Rhizobacter, and Rhodococcus. Scabs is also caused by the bacteria. Bacterial scab plant disease primarily infecting the belowing ground parts of the plants, roots, and the bacteria. The typically crocker mainly the common scabs are caused by the small number of the streptomycosis species. Vascular disease of the wilts. Vascular wilts caused by the bacteria primarily. Effect of the herbaceous plants such as the vegetables, field crops, ornamentals, and some tropical plants. A casual pathogen enters the multiplies and moves through the xylem vessels of the host plant and interferes with the translocation of the nutrients and water of the producing them. The pathogen will often destroy parts of the cell wall of the xylem vessels, resulting in the pockets of the bacteria, gums, and the cellular debris. The symptoms of the bacterial wilt disease including the wilting and death of the above ground parts of the plant. In some cases, bacterial ooze tips outgrowth of the stomata and cracks into the surface infected leaves. Some embodied organisms causing the bacterial vascular wilt disease are the monospecies, Corynebacterium, Erwinia, Pseudomonas species. Next, paddy blight, bacterial blight of rice is also called as the bacterial blight of rice, caused by the Xanthomonas is one of the most serious diseases of the found the worldwide, and is the particularly destructive in Asia during the heavy rains of the monsoon season. Symptoms: bacterial blight is the vascular diseases. The symptoms are the symptoms are visibly at all stages in the plant development and all parts of the rice, except the root. It has the three significant symptoms: leaf blight, pale yellow, and crishac. The leaf blight phase, the symptoms develop mostly on the leaf blades, leaf sheath, and sometimes on the grains. Next one is the mainly the leaf bacterial blight is the cause of Xanthomonas variegae species. The bacterium is the gram-negative bacteria, non-sporing forming, short rods with the round ends, measuring 1.28 to 0.1, 1.1 micro, monotrichous polar flagellum. They are the single, occasionally in pairs, but not in the chains. Is the aerobe. The optimum temperature of the growth is between 25 to 30 degrees. This is cycle and control. The control by use the balanced amounts of the plant nutrients, especially nitrogen. Avoid the excess of the doses of nitrogen. Apply nitrogen. The split dose is the 50% basal, 25% the tillering phase, and 25% of the chemical initiation phase. Ensure the good drainage feeds in the nurseries. Use only disease-free seedlings and the careful handling of the seedling during the transplanting. If fields clean, remove weed host and the flow under the rice suitable straw, rice cotton. Follow the fall of the fields to dry in the order of the suppressor disease agents in the soil and plant residues. So disease-free seeds are the sorry or soy. Use the resistant variety, the Ajaya IR64, Sona Masuri, Sona Radha, Janaki Sujata, PR 4141, Prasad, Gobind, and next one is the Pandan 4, IR20. Chemical control of the bacterial blight. Chemical control is largely effective in the minimizing the bacterial blight. Seed treatment are the with bleaching powder and zinc sulfate. Seed treatment soaking seed in the 0.1 gram streptomycin. Seed soaking for 8 hours of the agromycin and the vegetable terrasen followed by the hot water treatment for 30 minutes at the 50 to 54 degrees centigrade. Citrus canker, next another disease, the citrus canker is also caused by the bacterial diseases. Citrus canker series is a bacterial disease worldwide. Destruction occurring the wherever the citrus grown. The disease causes the damage in India, China, and Japan and Java. Mainly this pathogen initiate the cell canker disease. The number of citrus species on stems, twigs, and leaves and fruits, causing the leaves to drop and the fruit fall to the ground of the before ripens. What are the symptoms of the citrus canker? Citrus canker can be serious disease at the rainfall and warm temperature are frequent during the periods of the shoot emergency and the early fruit development. The disease affects the all above the ground parts of the tree, but most successfully are the leaves, twigs, and the fruits. Citrus like first like disease, the less disease and the cabbage spots of the small cankers appear in all the above ground parts of the plant. The trees are however not commonly killed. The odor lesions become the croaky and the brown, sometimes the purplish with the rash in the margin of the sunken centering, assuming the cankerous appearance. The canker lesions contain the pathogens in millions. Casual organism of the citrus canker. Bacterial pathogen, the Xanthomonas citri, is now also called as the Xanthomonas campestris or the Xanthomonas citri. The bacterium is the short, the motile rod, and with the single polar flagella. It lacks the endospore formation. It is a gram-negative bacteria surrounded by the mucinous capsule. It forms the chains. The climate factors which favor the disease are the mild temperature and wet weather. The most suitable range of the temperature appear to be 20 to 30 degrees centigrade. This is the disease cycle of the citrus canker. The first one is the Pachylacillum active phase, uh, dissemination by the wind and rain and insects, post-infection through the wounds and the stoma in leaves, stems, and the fruits. These are the secondary cycles like the first. The dormant phase is the fermenting in the affected boost tissues. These are the main control by the eradication, pruning, and next one is the spraying. These are the control of the citrus canker diseases. Next one is the viral disease. Some of the viral diseases. All viruses are the obligate intracellular parasites. Do not have the molecular machinery to the replicate without the host. Viruses are not active outside of their host. Viruses cause many important plant diseases and responsible for the losses in crop yield and quality in all parts of the world. Viral diseases of the plants exhibit the variety of symptoms when no sign of the infection can be detected. Infection is said to be latent. Mosaic diseases, some of the viral diseases in plants, mosaic. The most common symptom due to the viruses infection, mosaic. Interruption of the various degrees of the chlorosis. With the normal green color of the leaf results the mosaic with the yellow and the green color. When there is the uniform reduction of the chlorophyll, the symptom is the referred as the yellow type. Vein clearing and vein bending. The symptoms of the systematically infected leaves. Vein clearing occurs earlier in mosaic or chlorosis of the tissue in our immediately adjacent to the veins. Vein bending consists of the broader bands of the green tissue appearing along the veins. The two symptoms may be the short and the duration. Ring spots. The spots appear in the localized spots and the consist of the various types of the chlorosis and necrosis. They may be circular chromatic areas. Leaves are the called as the chlorotic ring spots and necrotic ring spots. Necrotic may appear in the alternate with the normal green. The centers of either type of the spots may be eventually become necrotic and are also called as necrotic ring spots. Necrosis is the death of the cells and the host tissues. It appears in the various forms. Usually, it is the restricted to the small areas on the leaves or the spots on the stem. In the formal case of the chlorotic tissue may break down and the necrotic spots appear. In seven necrosis, there is the rapid killing of the terminal bud, branch, wilt. In certain viruses infected plants, the whole outer walls of the leaves drop and become the yellow, stunting premature defoliation. If the viruses disease, generally the hypoplastic subnormal cell production of the response to the attack of the pathogen. It results in the under the development of the host spots. The host plant remains the stunted with the shorter internodes of the smaller leaves and the dwarfing of the host plant. The viruses infected plants are the most often stunted in the kill. Outer growth, flower breaking, fruit abnormalities, symptoms such as the molting, sour crop may develop the fruits of the certain viral infections. Rice tuber, RTD. These are also caused by the viruses. Rice Tungro bacillor farm viruses RTBV and Rice Tungro spherical virus RTSV. Now the disease occurs severally and one of the most damaging of the distract diseases to rice production. Rice Tungro is the very serious disease. Most of the affected the rice crops. The symptoms of the most conspicuous symptoms of Trungro are the stunting of the plant and show the various shades of the yellow to orange discoloration of the leaves. The symptoms may vary according to the age of the plant, rice variety, and the virus strain. Yellowing of the tips of the young leaves is the first symptom to appear, and this may be include the intervenial fluorosis, the chlorotic molding, casual organism of the disease caused by the simultaneous infection of the two viruses, rice tungro, bacillariform viruses, RTBV, rice tungro, spherical viruses, RTSV. The both the viruses are the transmitted by the green leaf opus, nematode the virusensis, RTBV, described by the Badna viruses. There's the bacillary form capsid measuring the 130 nanometer and 30 nanometer. RTSV, Vaccia viruses. There's the iso diameter capsid measuring the 30 nanometer diameter and the made up to 2 to 3 species of the core protein. It contains the single stranded RNA. It's a cycle. Control of the rhizome growth disease, control method, cultural method, sanitation, transplantation of the time, location of the seed bed. These are the cultural methods. Chemical control, just after the emergency of the seedling to the application of the Potax, seedbed keeps the area free from the vectors. Spraying of insecticides like the carbofuran and the cypromethrin, it gives the significant reduction of the vector population. Application of the carbofuran at the rate 1 kg of hectare. Next one is the tobacco mother. The rice tumor is the affected by the rice. The viruses diseases. Next one, tobacco mosaic viruses. Tobacco virus is also called as the nicotina virus. Is the best known of the all viruses disease and the found in the susceptible plants of the tobacco are grown. It causes the enormous losses in the tobacco crop, which include the poor quality and reduce the yield. The mosaic virus can attack about the 200 species of the herbaceous dicotyledons plants, including the many vegetables, flowers, and weeds, which is the most important tobacco and tomato, but it does not affect any monocotyledons plants, only affect the dicotyledon plants. Symptom of is the systematic mosaic type. The first common symptom is the downward curling of the and destruction of the youngest leaves, accompanied by the chlorosis in the young tobacco plants. A number of dark green spots appear as the leaves enlarge. These spots develop into the irregular blister-like the areas, whereas the other parts of the become chlorotic. The light and the dark green patches give the leaf of mosaic pattern. The other symptoms which follow are the various types of the leaf malformation. The old leaves may develop large yellowish blotches without any sign of the molting. The plants remain the dwarf unstunted. Casual organisms, tobamoviruses. Tobacco is also known as the nicotina viruses. These are the first viruses to be discovered in the Ivano whiskey in 1892. Tobacco particles are the rod-shaped viruses. The virion measures 18 into 30 nanometers and weights 39 million daltons. The capsid is made from the 2130 130 proteins. Each subunit consisting of the 150 amino acids, which assemble around the viral single-stranded RNA. To form the helical structure with all of the central cavity of the 4 diameter. The single-strand RNA consists of the 6,400 nucleotides. This is cycle, active phase, and dormant phase. Active phases are mainly inoculation of the seedlings and transplants during the handling. Next, systematic mosaic. There are the disease cycle. In the disease cycle, the tobacco mosaic viruses. This is the disease cycle tobacco mosaic viruses. The cycle active phase, dormant phase. Active phase mainly is the viruses dried leaves and the living blows. Dormant phase is the penetrating the virus plant and destroying the plant. One of the common control methods of the TM is sanitation, which includes the removing of the infected plants, washing hands in between each planting, and restrict the access to the area. Growing viruses free plants, propagate plants via seeds rather than the vegetative or crop rotation. Growing of the resistant varieties, removing of the all weeds during and after the growing season. Place of the tools in the disinfect of the solution for the at least 10 minutes and the rinse thoroughly with the tap water. Discouraging of the tobacco by workers. Tungro viruses, ATM is the both are the affected viruses diseases. Okay. Rice tungro viruses is the mainly effect on the rice plants. And as a TMA viruses, they can affect of the tobacco plants. Some of the Solanaceae family members, tobacco and tomato. Next is the mycoplasma diseases. Mycoplasma are the eubacteria of the class moleculates, the group of organisms the phylogenetically related to the gram-positive bacteria. Their characteristic features reside in the small size of the genomes. The low guanine and the cytosine G plus C is the low quality. These are the gram positive bacteria, and low level of the guanine and cytosine content in the genomic DNA. They are the simplest and the smallest known organisms capable of growing the cell, pre-laboratory cultures and media containing the cereals. Mycoplasma lack definitive cell wall and are the characteristically delimited by the distinct, soft, flexibly trilaminar unit membrane of the lipoprotein. These characteristics makes them the naturally resistant to the antibiotics, that is the target cell wall synthesis. They can be parasitic or saprophytic plant pathogenic mycoplasmas, the responsible for the several hundred diseases. Reproduction of the mycoplasma has not yet been conclusively provided. They can reproduce by the binary fission, by the formation of the spores, elementary parts. Mycoplasma structure, these are the mycoplasma structure. This is the mycoplasma structure. Okay. They're containing the ribosomes, some of the soluble proteins, protein membrane, metabolites, DNA. Is a membraneless model. These are the membrane mycoplasma is the membraneless model. Symptoms of the mycoplasma disease in plants, phylloid disease. These are the abnormal development of the flower plants in leaf structure. The affected plants show the reduction of in leaf size, short-ended the internodes, general stunting, abnormal branching, resulting in a plant malformed beyond the recognition. Pillow causes the effect of the plant become the partially or entirely sterile, with unable normal produce the flowers. Spike diseases. Spike infected plants show the extensive deterioration of the root system. Spike disease affected trees die within the 1 to 2 years. Affected little leaf diseases include the shortening of the petioles and production of the leaves, which are the extremely smaller in size, giving bushy appearance. Petioles are also shorter than the leaves. Grassy shoot disease plants show the production of the numerous thin, short, lanky, abloyant tillers the base of the affected plants, which give the typical grass-like the appearance, hence the name grassy shoot disease. The leaves the affected plants and the produce the chlorophyll. Little leaf of brinjal. The most serious disease is the brinjal solanum melangina in the areas of the cultivation cause the qualitative as well as the quantity loss. Casual organisms of the little leaf of brinjal. Little leaf of brinjal mainly are the affected by the some of the Solanaceae family members. Casual organism this is caused by the plant pathogen moleculate phytoplasma, earlier known as the mycoplasma like the organism MLO, MPLO or MLO. Control this is the disease cycle mycoplasma can affect plants in all the growth stages. Probably during the season of the brinjal crop, the casual agent survives on weed host and from there is the transmitted to the main crop crafting through the insect vector. Control of the mycoplasma diseases, the little leaf of brinjal, adopting the sanitary measures which include the eradication of the susceptible volunteer crop plants from the previous planting, use of the barriers and drop crops, early removal and the destruction of the infected plant. The sowing time can be adjusted to avoid the main flights of the bait leaf gopher. Cultivation of the disease-resistant varieties are the principal means of the management of the diseases. Chemical control, spraying the malothanin and the thiamethoxam accepted and the thiocyclone hydrogen oxalate. The grass shoot disease of the sugarcane. The grossy shoot disease the sugarcane across the most of the Asian countries, affecting the sugarcane production to considerably level, and these loss are the higher in the rotten crop. The disease caused by the phytoplasmas in India. Disease occur all the sugarcane growing starts after the resulting of the 100% loss of the cane yield in the sugar production. Symptoms: the infected sugarcane plants show the proliferation of the tillers, giving the typical grassy appearance. Hence, the name grassy shoot disease, the disease that displays the different types of the symptoms at the different stages in shares. Casual organism candidates the phytoplasma as affected one of the destructive pathogen in the sugarcane, Saccharum obscinarum, candidatus phytoplasma. Control cultural method growing the resistance varieties Co 86 to 49, CoG 93076, and CoSE 22. Avoiding the routining of the gross issue disease incidence is the more than the 50% in the plant crop. Physical method pre-treating of the sets the at 50%, 50 degrees centigrades. Physical method pre-treating with hot water for 55 degrees for 8 hours. Chemical method spray the spray dimethanate 1 ml in the 1 liter water is the control insect vector. Spray pesticide dimethane. All are the uh, viral diseases, some of the bacterial diseases, some of the viral and mycoplasma diseases. These are all are the block one. The major disease of the in crop plants. Actually, major crop plants, especially rice, wheat, okay, sugarcane, all are the major. The next one is the major insects, pests, and the parasitic plants of the crops. Cotton, the commercial crop, is the backbone of the textile industry in the world. China is the highest cotton produced in the country, ranking the first position of the followed by the India, occupying second position. And United States of America is the third largest producer in the country. The adult foreways are the brown in color with white markings. The hindwings are the white color with the brown patch along with the margin. The egg larvae laid the messes, which is appear golden brown in color. The larvae are the pale greenish with the dark marking. The larvae exhibit the gregarious behavior in the early stages. For cotton aphid control, the management chemical control is very effective in the adding the cultural and other control methods. Cotton seed treatment with the imidacloprid. The successful chemical control can be achieved by the spraying imitated 200 SL in the methyl dimethyl or the dimethyl. All are the some of the major insects and the pest parasites. Insect pests also, for example, fruit borer, Heliocorpia, Armiger, Fabiner, and the pink ballworm, Pectinophora gossyliferus, Sanders, spotted bollworms, Hervius vitiela. Next one, the cotton stem will, perhaps. The next one is the stem borer, Spintora grossier, leaf roller, Selefia dergota, tobacco cutworm, Spinora lilios, and the ash Willis, Malurus under central is the Marcellus, Debrucius, the cotton aphid, Apis, Gosephia, glow, thrips, and red cotton mysterious, the Cinclorus, dusky cotton, Oxyrus, the Apis, the all are the tobacco, cutworm, and the cotton aphid, the selected the detail. When tobacco cutworm, the subroad, the lituria, damage of the symptoms, management, the cotton aphid, the Apis, Gosephia, description of the insect pest. The cotton aphid, the adults are the greenish brown small insects with the soft body. The late as well as the apterous forms are the present and the females multiple pathogenetically and vivipariously. A single female may give the birth 8 to 20 nymphs day, which is the become the adult about the 7 to 9 days. The aphid nymphs are the found in the under surface of the So these are all are the some paddy and the storage phase. We discuss the next class economic importance of the paddy, storage insect phase. Most of these are the angiosperm parasitic plants, fruit parasites, and next one is the plant pest management.